Hello, everyone, and welcome to Integrating Plasma Proteomics to Identify Tuberculosis-Specific Diagnostic Biomarkers. I am Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink Proteomics. To learn more about our sponsor, please visit olink.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and then click Send. We'll try to answer as many questions as we have time for at the end of the presentation. If you have any technical issues as well, having trouble seeing or hearing, you can also send those in the Ask a Question box and we'll address those to you. I'd like to now welcome our speaker, Dr. Hannah Schiff, NIHR Clinical Lecturer in Respiratory Medicine, University of Southampton. For a complete biography, click on the biography window on the left-hand side of your screen. Dr. Schiff, you may now begin your presentation.
Thank you very much, Matt. And hello, welcome everybody. And thank you very much to LabRoots and Olink for the opportunity to present the research that I've been doing in Southampton, UK. Using Olink technology and mass spectrometry to identify TB-specific diagnostic biomarkers. So this is very much a case, clinically orientated research. Just first a little bit more about TB. So TB is a disease that's been around for thousands of years. It's caused by a mycobacterial infection, Mycobacterium TB. It's spread by coughing. And as you can see here on the left-hand side with some images, it's spread by coughing with respiratory droplets that are then inhaled by other people, that then sets up the infection predominantly within the lung, although TB can affect other parts of the body. But it's transmitted by coughing, and I'm a pulmonologist and lung doctor and interested in diagnostics and interested in trying to diagnose this lung infection and see how we can prevent transmission. And TB sets up quite significant destructive pulmonary pathology, as you can see in the images on the right. You can see, if you see the lungs macroscopically, the big holes in the tops of the lungs. You can also see cavities if you look on plain film chest x-ray and CT scan. And if you do a PET CT, with radioactive tracer that highlights metabolically active tissue, you can see that the outside of this cavity lights up, which is reflective of high levels of inflammation and metabolically active tissue. And it's these bacteria that are contained within these cavities that the patient then coughs up, and they're inhaled by other hosts. And how it's transmitted depends on the number of bacteria TB patient expelled, also environmental factors, where that exposure happens, the concentration of TB that's expelled, enclosed spaces, poorly ventilated areas where people are packed tightly together, all increase risk of transmission, and how close you are to someone that's coughing up TB, how frequently, and the duration of exposure all increase the probability of transmission. And then once you inhale the TB bug, the disease that it sets up depends on many host factors and how susceptible that individual is. So TB, as I'm sure you know, is a disease of global significance. This slide shows data from the latest WHO Global TB report published this year. And then the graph on the left shows the difference between case notifications of people newly diagnosed with TB with the estimated number of incident cases, which is in the blue line at the top. And essentially, TB affects over 10 million people a year, 1.3 million people die, and then this big gap in the middle, as highlighted by this arrow, shows that there's actually a case detection gap of approximately 3.1 million people globally. So 3.1 million people are coughing up TB over other people carrying on driving the pandemic, and that are not being picked up by current diagnostic methodologies. And the map on the right shows that the highlighted areas where this diagnostic gap is largest. You can see there by the pink circles. And these misdiagnoses increase TB transmission, the chance of getting significant lung disease, with delays of diagnosis and ultimately mortality from the disease. Currently available diagnostics centre on detecting the mycobacterium tuberculosis itself, so detecting the bacteria. And for lung disease, that requires a sputum sample, which some patients can't expectorate or are difficult to obtain. And then once you get it, you need to examine the bacteria under the microscope. And then samples can be sent for culture, which take up to several weeks for an answer. Meanwhile, before that diagnosis is made, people can cough TB over other hosts. There are more rapid molecular tests, such as a gene expert test, which tests for TB DNA in the sample. Again, these can be performed on sputum samples. And there are urine tests that detect lipoaruba which is in the mycobacterial cell wall. But all these tests have limitations. There's need for a sputum, there's time to the result, there's the cost per test, and the need for specific for laboratory diagnostic personnel. And the sensitivity can be poor in some of them. So you can have active pulmonary TB, but all the diagnostic tests can still be negative. So my research that I've been undertaking with the TB research group in the University of Southampton has been investigating a new diagnostic approach that utilises plasma proteomics, with the hypothesis that are the proteins that are released by this lung inflammation and cavitation actually clinically useful diagnostic biomarkers. So in order to investigate this, I optimised a mass spectrometry protocol that's developed in the University of Southampton that closely profiled the plasma proteome from 11 TB patients and 10 healthy control patients from a mixed Peruvian and South African cohort. And these were all male patients, carefully clinically characterised. They were all microbiologically diagnosed tuberculosis. They were all HIV negative. They were all male. They all had abnormal chest x-rays with signs of destructive lung disease. And they didn't have any other confounding factors, such comorbidities that might affect the plasma proteome, such as diabetes, other diagnoses, such as significant liver disease, renal disease, malignancies, for example. And this plasma proteomic protocol works for a non-depletion technology. So many other mass spectrometry approaches to profiling the plasma proteome deplete out the most highly abundant plasma protein, which is albumin, which makes up nearly 50% of the blood protein, to try and reduce the complexity of the plasma that's being analysed. But this has been shown to co-remove potential other molecules of potential diagnostic utility by this immunodepletion protocol to start with. So this plasma protocol was particularly good because it basically worked on serial orthogonal plasma separations to try and separate the plasma proteins out initially by size exclusion chromatography into loose plasma segments of molecular weight. And then we went on to trypsin digest the proteins and label them, and then expose them to orthogonal serial chemical chromatographic separations, which separate the peptide fractions out. And then each peptide fraction was individually loaded into the mass spectrometer for analysis. That all the proteins were tagged, so that doesn't alter the way the protein flies in the mass spectrometer, but the tag can then be married back up. It changes on the secondary fragmentation. You can then identify which protein which patient the protein came from. So that is quite an extensive lab protocol. The protein was separated into four plasma segments, and there were 3 different experimental runs, which left 12 individual experiments, which take about 2 to 3 weeks to run. So it's very extensive, painstaking chromatographic separation, but has elucidated a really carefully characterized and really detailed and large plasma proteome. So in this slide, you can see the plasma proteome without any differential expression analysis. This is unsupervised data analysis. So on the heat map on the left, you can see the fully quantified plasma proteome. So the whole experiment identified over 2,300 proteins. But in order for full data analysis, you had to get a quantification result in each patient. And that was 594 proteins. And so the heat map shows the 594 proteins, which is the complete quantified plasma proteome. And you can clearly see with unsupervised data analysis that the patients cluster into healthy controls and pulmonary TB. And if you've got really good eyesight, their samples are also marked into the different ethnic backgrounds of the cohorts, and their samples also cluster to the different ethnic background of the patient, which is another really interesting finding. You can see on the right that with principal component analysis that the protein abundances from the quantified plasma proteome also separate nicely in principal component space. And this was before any differential protein expression analysis. So hopefully you can see that slide. So this is the bioinformatic approach to the biomarker identification that I took. So there's a lot of detail on this slide, but I don't want that to be discourage anybody. It's basically to say that there were those 12 plasma segments, and then I combined the plasma segments bioinformatically to elucidate the full plasma proteome. And then I took all of that data through some complementary bioinformatic pipelines to identify differentially expressed proteins. So I used a linear modeling approach for proteins that were mathematically differentially expressed, adjusting for multiple comparisons. And I also used something called WGCNA, which is a whole genome correlation network analysis, which is a systems biology technique that was adapted from genomic data analysis to proteomic data analysis to identify clusters of co-related proteins that correlated with the condition of having TB compared to controls. And then proteins that came through all three diagnostic pipelines that were in the middle of this Venn diagram were then taken forward as candidate diagnostic biomarkers. And that's also summarized in the flowchart to the right-hand side. So this is what the Venn diagram looked like. So this approach identified 118 differentially expressed proteins that were identified by these complementary bioinformatic pipelines. And if you plot them on a volcano plot, these are all the ones that are statistically significant. There were 56 upregulated proteins and 62 downregulated proteins. And you can see in the upper outer quadrants, they're the proteins with the highest log fold change and the highest Z-score, which is a correlation within the module identified by WGCNA. So you might hypothesize that these would be the best candidate diagnostic biomarkers, and you'll see that CRP is on there, and CRP are probably common to many of you in the audience, and has been explored as alone as a diagnostic biomarker, but it's very poorly sensitive in HIV negative patients. So I also then asked the question, are these proteins that I identified through differential protein expression actually reflective of the biological processes that are happening in the lung in TB? And I did some gene ontology analysis to explore that further and try to identify the biological processes that are enriched for these proteins. So this chord diagram shows the highest, the proteins with the biggest log fold change, either upregulated or downregulated, and links to their enriched biological processes. And you can see that there are important processes in there like proteolysis and extracellular matrix organization, which biologically are key to the processes. So we think we've captured a very a good representation of what's going on in the lung and pulmonary TB in the blood plasma. This CNET plot is a very similar way of trying to illustrate the same point. This is linking the upregulated and downregulated proteins to significantly enriched biological processes. But in this diagram, you can see a little bit more of a network and which the directionality of the differential expression. So then I was left with the nice problem to have is then trying to validate the differential expression of these proteins. So we had this large data set, this great plasma proteome. We've got our 118 differentially expressed protein candidates. We think we've got a proteome that's representative of the events that are happening in the lung and pulmonary TB. How do we validate this? And we're interested in antibody-dependent validation methodology. And we have done some validation, including ELISA and Luminex, but you very rapidly encounter problems with multiplexing and sample volume, and discovered the Olink technology that uses proximity extension assay. And this relies on dual antibody binding in close proximity of the protein. And these antibodies are oligonucleotide tagged that when they bind in close proximity will hybridize, and that signal can then be amplified up and marked with DNA barcoding of each protein and sample. And then with a sequencing or a PCR readout, you can read out a signal proportional to the target protein amount. And I discovered that 55 of the 118 proteins I was interested in were present on the PEA Olink panels, the cardiometabolic and inflammatory 2 panels of Olink. And this uses a DNA sequencing readout. And the output is something called normalized protein expression. So it's a relative quantification, still similarly to the mass spectrometry. So we then took these, we wanted to also validate these markers in a different patient cohort. So I had access to a UK collected diagnostic cohort called MIMIC. These are actually serum samples rather than plasma samples of a mixed sex and mixed ethnic background, and tested these markers, so these 55 of the 118, using these proteomic PEA panels in patients with in healthy control patients, TB patients, and also a really key comparator group of people with other respiratory tract infections. So these were people who had other lower respiratory tract infections that were microbiologically confirmed diagnoses, so may present similarly to patients with TB, but actually have a different underlying diagnosis. We're looking for markers that could were not only raised in TB, but offered a degree of specificity separating out TB from other respiratory infections. So you can see here on this slide that there are a lot of proteins that we managed to validate in this group of 55. So my initial analysis just concentrated on the 55 proteins, although we measured about 700 proteins on these 2 panels. So I could see that 34 of them were significant between TB and healthy controls, and 18 were significant between TB and other respiratory tract infections. But actually there were 4 that were of particular interest, which are shown in these box and whisker plots, which show that there are 4 proteins that showed a really high degree of TB specificity, as in they were increased in TB, but not in the comparator conditions. This was a really exciting finding, because often all these proteins are host protein markers, and whilst can be sensitive, specificity can be challenging. So this was really exciting. I then took forward these proteins and tried to investigate combinations of proteins that might be diagnostically useful. So I did some receiver operating curve characteristics and identified a CombiRock package that you can use in the R programming language to identify combinations of biomarkers and investigate their diagnostic potential. And I discovered that there was a 6 protein panel that you can see here on the rock curves that, when you plot individually, have pretty good area under the curves, but actually when you combine them using logistic regression and then plot the receiver operating curve characteristics, show a really high degree of sensitivity and specificity. So you can see in the truth tables under the TB and healthy controls that this 6 protein panel can differentiate controls with a 90% specificity and an over a 90% sensitivity, which is really excellent performance. And then if you do the comparison between TB and other respiratory tract infections, there was also a maintenance of sensitivity and specificity. And this is really exciting because published target product profile criteria for a non-sputum based TB diagnostic. And that is a sensitivity of over 90% and a specificity of over 70%, of which they both met. We then took this 6-protein panel forward and validated it in a second clinical cohort. So this was a cohort that was collected in South Africa. This was plasma samples, again with the same groups of controls, TB and other respiratory tract infections. It was a mixed-sex cohort, all exclusively Black ethnicity, and again used the same proximity extension assay panels to investigate the protein expression. And you can see when we just looked at those 6 proteins of this really highly performing diagnostic panel, again, there is this maintenance of a significant degree of TB specificity, which was really exciting to see. And then when you combine the proteins into the 6-protein panel, again, there's really good maintenance of sensitivity and specificity values. There was a little bit of a drop down in sensitivity versus the healthy controls, but interestingly, between the TB and other respiratory infection, these sensitivity and specificity was really excellent. And the other respiratory infection group in this cohort were patients that presented with symptoms similar to TB, but were negative for TB on subsequent diagnostic testing. But because of the resource limited nature of the healthcare system, they didn't have an underlying confirmed microbiological diagnosis. But the important thing is that there were patients presenting like they had pulmonary TB, but didn't when you tested them. So it's a very clinically valid comparator group. So this slide just presents a summary. We published the results in JCI Insight earlier this year, and this is the graphical abstract from that paper, which tries to summarize how we use the integrated proteomics approach to identify these TB-specific diagnostic biomarkers. And it's shown that through this hypothesis-directed approach, but using unbiased technologies, it's identified disease-specific candidate diagnostic biomarkers. And a 6-protein panel can differentiate pulmonary TB from controls and comparator conditions. And really excitingly, as the Olink panels work through antibody-dependent methodology, they can be validated. They're not only found by mass spectrometry, but can be validated in something that uses antibody recognition, which has really exciting potential for further development as a diagnostic that might be able to be brought closer to the patient eventually. So are we moving closer towards a near patient test for pulmonary TB? Well, I think there's definite potential. Leaders in the field, as you can see from this reference here, have advocated the steps forward. So moving from more microscopy-based diagnostics to molecular diagnostics, moving away from centralized and lab-based tests to decentralized point-of-care tests, potentially integrating single disease testing into multi-disease testing, which my talk doesn't really address, and moving away from sputum-based diagnostics to non-sputum-based samples, which we have ticked that box. And also really what the patient need in TB is in lower resourced countries, and actually taking diagnostics out of healthcare facilities closer to communities and homes and patients is what's required. And something that's lower cost, and actually a near patient diagnostic needs to be something that works if you're taking it out of a lab environment potentially, which this graphic on the right hand side, it would be the dream. We've all had experience of lateral flow tests in the COVID pandemic, And if you can imagine something like a lateral flow, it's low cost, cheap to manufacture, doesn't require a cold chain, doesn't require electricity, doesn't require a skilled operator to manage. You could see if you had the right set of diagnostic biomarkers, you could take it, and it could be translated into something like this. You could take it out into communities to try and find those missing millions of people, to at least triage them into a better diagnostic, confirmatory diagnostics. And such active case finding. Sorry, I think I haven't skipped on the right slide. Apologies for that. So I was talking on the wrong slide. Many apologies. Hopefully you've got the right slide now. So active case finding with such a test as the lateral flow test on the right-hand side could therefore close the case detection gap that's fueling the pandemic. So what's next for the research? So we have further data analysis to work on interrogating the PEA data sets to explore other diagnostic biomarkers. I'm also interested in how other host phenotypic characteristics might be able to be combined with diagnostic biomarkers to enhance biomarker performance, because we know that there are a lot of host factors that impact on the clinical presentation of lung disease. We'd like to do much more validation and testing in larger clinical cohorts in larger numbers, and also development of a bespoke PEA assay panel. So Olink have several explore panels, proteomic exploratory panels, which is what I've used in my research, but also they have smaller panels that offer absolute quantification. So in an iterative process of validation and narrowing down the further biomarkers, we could work through via that step into a diagnostic. And ultimately to try and translate it into a diagnostic device suitable for use in near-patient settings. And that would, if we arrive at that step, that could then be prospectively tested in areas with high disease burden to see if it can make an impact on finding these cases. Right, I'll try and, sorry, I'm having a few problems to getting the right button to turn the slides, but this is my acknowledgement slide. Thank you very much for your attention. I couldn't have done all this work. I have to acknowledge the TB research group in Southampton, particularly Professor Paul Elkington and Diana Garay-Buqueri, who pioneered the mass spectrometry protocol, and my collaborators in Peru, originally in South Africa, and also Paolo Piazza in the Technology Platforms Group, who's the service provider for the Olink panels, Dr. Spiros Garbis and Dr. Antigone Manasupoulou, who I worked with on the proteomics, and ultimately all the patients for providing the samples for analysis. Thank you very much for your attention.
All right. Thank you very much, Dr. Schiff, for your very informative presentation. We'll now start going through the live Q&A. So if you do have any questions that you haven't sent through yet, go ahead and put those in the box now. Like I mentioned, we will try to get through as many of them as we have time for. So for our first question, let's go with, does the panel distinguish between active and inactive TB?
Yeah, great question. So far we've only tested this panel between active pulmonary TB, and we haven't done any investigation of this particular panel in latent TB. We have done some smaller validations using ELISA and Luminex in latent disease, but there were different markers that are presented in this work.
All right. Let's go with, can you maybe comment on the approximate concentration of the 6 TB proteins you identified? Order of magnitude would be sufficient.
Yeah, also a really good question. It's all relative quantification, and the MPX scale is a log scale. So actually orders of magnitude are potentially quite large, but obviously we've published the data in the paper, so I'd happily refer you to the paper. And I'd be happy to be approached to look at the data in a bit more detail if there was anything in particular that you would like to know. It's all relative quantification at the moment. We're obviously interested in absolute quantification, and that will come in next steps.
All right. Let's go with, how stable are these markers in the serum or plasma? And is there a difference or preference of serum over plasma?
Yeah, interesting question. So it seems to work, these PEA panels seem to work really well, both in plasma and serum. And some of these cohorts were collected some years ago, I mean, almost 10 years ago. So we've analysed plasma that's been in the -80 for almost a decade and got these good results. So actually pretty stable in these samples so far. We have tried some, not necessarily stability of these particular proteins, I think, but have tried some validation on certain proteins that are actually quite hydrophobic, which has caused problems with identification via antibody-dependent methods. Not so much in this PEA, but in other, we tried quite hard to measure a surfactant protein, which was very, very hydrophobic and had problems with antibody binding, and we could not get any assays to work. And I think we put that down to the hydrophobicity of the protein.
Thank you. Similar as before, is it possible to discriminate between active TB and latent sublatent?
So I'd love to be able to test these markers in latent and active TB. At present, the analysis plates for the PEA panels are limited to 88 samples, and you can do multiple plates with bridging samples, but at present we've only done single plates. So in order to maintain the power of our analysis for comparison, we only had 3 clinical groups per plate, so had to forego any latent testing so far, but be really keen to see how it performs. I think certainly from other proteins, I mean, you don't know until you validate is the ultimate. Everything has to be tested then. We haven't found any predictors of what will work better than others, assuming you can get an assay working, but we're very keen to test in latent disease in the future.
Let's go with, are p-values in the volcano plot and other plots FDR adjusted p-values?
Yes, yeah, they're all adjusted for multiple corrections, and the volcano plot is a slightly unusual volcano plot because it had different things on the X and Y axis, because it was combining 2 bioinformatic analyses. So yeah, they were all FDR corrected that was displayed, and there isn't an FDR cutoff line on the graph like in some standard volcano plots when you just do the log fold changes.
Okay. I have a bunch coming in here. I wonder if you have partitioned data into train and test from making ROC.
Yeah, also great question that I would like to explore. So at the moment, we haven't done that, but I'm looking into machine learning approaches to do that. We're slightly limited by the current sample sizes, but be really happy to talk to anybody about approaches that anyone's taken and discuss that, because we're really interested in doing that on the data, because I'm sure that there's more diagnostically useful proteins to be extracted from the data that I have.
Okay. Let's do what technology is used to diagnose tuberculosis, and also discussed about the treatment of TB?
Yeah, thank you. So I think I touched a little bit on the technology. So I mean, diagnostic technology hasn't changed for a long time. The mainstay of diagnostics is examining the patient's sputum for mycobacteria under a light microscope and then culturing it. And and because Mycobacterium TB takes a long time to grow, this can take weeks, if not months sometimes, to get a result. They're the basic diagnostics. There are obviously molecular diagnostics, which I think I touched on. So looking for essentially nucleic acid amplification tests, NAT tests, which are looking for MTP DNA. That's such technologies as the Gene Expert and other things. But again, they're all the diagnostics are great if you get an answer, but they're all really limited by poor sensitivity or cost or time to result. And actually, where the diagnostic sits is sometimes affecting, the diagnostic rates that you get. So there's a massive gap, as I showed in one of my earlier slides, between, even though these new diagnostics, where they've been rolled out, they've not been shown to affect mortality of patients, and are clearly not closing that case diagnostic gap. So there's definite need for innovation to try and identify these patients earlier, which has individual benefits, population benefits, transmission benefits. Hope that answers the question. Sorry, I don't know if there was anything a bit more that you wanted to ask.
And if they do have any clarifications, they can send those in, and we'll watch for them as they come in. Are any of these proteins affected by the BCG immunization status of the patient?
Another great question. I don't know for sure because I don't have all that data, but certainly the South African cohort are unlikely to have had BCG vaccinations. But that's a good question. I don't think so, based on the data that I've got, but I would say that we don't really have that information about the patients in the studies so far that would be able to answer that for sure, but that would be an interesting point to look at.
Is there any specific criteria requirement for sample transportation, especially if the samples take more than 3 days to reach?
I think I'd need a bit more clarification on that. Let me see, sample transportation. So the research I've presented is all done on plasma or serum samples that have been collected and then stored at -80. The original mass spectrometry work was all done on the samples were fixed in 6 molar guanidine, which essentially arrests all enzymatic activity, so tries to freeze the proteome, if you like, in time so that you can then do the subsequent analysis. And then that 6 molar guanidine is all dialyzed off in the analysis process. The PEA assays were all done on -80 stored plasma or serum samples that were then thawed for analysis. In terms of, I think if there's questions about how if you wanted to analyze samples in this way using these techniques, certainly the PEA assays, they're commonly done on serum and plasma, but it's all very much in a research capacity and not a clinical capacity at the moment. What else would I say? I would say also that as you probably allude to, any delays in delays in transport or delays to analysis could introduce effects on the data. So technical effects, so the pre-processing effects on your protein analysis or differential expression analysis, and we try to minimize any sources of pre-analytical variation as much as possible, so that we're really just looking at the differential between disease and healthy, or disease and comparative disease.
Thank you. Are there plans to validate this signature to more easily accessible sample types?
Well, we think blood's pretty accessible compared to sputum. There are a lot of people that can't produce sputum, and it would be great if you had a urine diagnostic, for example. The caveat with that is that urine is a much more physiologically variable sort of body substance, and blood and plasma is very tightly physiologically regulated. So in terms of discovery work, that's why we chose to work on that. It would be great to know if some of the most exciting proteins could be detected in the urine. I think that sort of moves on to other questions that people might be asking about development on into other forms of diagnostics, lab diagnostics or in vitro diagnostics, excuse me. But that raises points about it's a host protein and has to reach a certain point. There has to be a cutoff, and it's not just a presence or absence or on off. So if it was a TB protein you're detecting in the urine, if it's there, great, it's positive. If it's not there, it's negative, largely. I mean, if it's not there, it doesn't mean it's not there. So we won't get into that discussion. But with host markers, there's going to have to be a cutoff. And actually, I think defining those cutoffs is probably going to be easier in the blood, and then you could then take that forward, perhaps investigate it in the urine. Sorry, I'm probably not. Let me know. Please get in touch with me if there's anything I haven't covered in the questions.
All right. What is the biggest challenge in adapting the identified biomarkers from the discovery phase into a clinical diagnostic tool, especially on a point of care test?
Yeah, I mean that would be the dream. I mean, the hurdles are multiple and big, but first of all, you need the right combination of markers, and then you need a device to put them on, and the translation would depend on the device. But I think we need to progress through iterative steps, as I alluded to. I think the sensitivity of the readout is a problem, because you're going to need you're going to lose sensitivity, and you're possibly going to need some kind of diagnostic reader of a device, or something that can do more than just naked eye looking at a lateral flow test. So getting the requisite diagnostic performance on a device that could be clinically useful, I think kind of boils down to multiplexing and sensitivity.
All right. Let's go with, is this useful in assessing the success of therapeutic interventions?
I'd love to know that. So the next follow-ons would be, you'd hypothesize if these markers are markers of active lung disease, you would hope that if you then treat the patient for TB and their lung disease gets better, that the markers would fall. And they could be used to assess treatment response, for example. Maybe if they've got multi-drug resistant disease, they would fall less fast, or they're not responding to the right antibiotics, they would fall less fast, and you could use it clinically to titrate treatment, change treatment to markers of response to treatment and potential outcome. And there's also a lot of interest in the field about post-TB lung disease. So actually, TB can leave patients respiratory very disabled, or with obstructive lung disease and fibrosis. So actually they might be good markers of predicting post-TB lung disease. So yeah, really interesting question, and yeah, would love to do longitudinal studies to find out.
Right, excellent. Did you have any information on Aspergillus status of tested patients? I apologize for mispronouncing that.
No, aspergillosis. As far as I'm aware, none of the patients had aspergillosis, but these were at diagnosis and didn't have any evidence of clinically or diagnostically, certainly in the discovery cohort of the mass spec, any evidence of aspergillosis. But it would be interesting, again, we've been asked that question about, could it differentiate them? I think in certain clinical scenarios, trying to work out whether someone has TB or aspergillosis is really important. As far as I'm aware, none of the patients in the study had aspergillosis.
And have you tested these TB-specific diagnostic markers in a TB-HIV co-infected cohort?
Not yet, but I've got grant funding to do that, to do exactly that. So we're going to test them in HIV co-infected patients. So hopefully have an answer in due course.
Excellent. Could sampling dried blood spots a good alternative for sample collection in the case of no lab freezers are not available?
Yeah, and we've definitely been considering that as a future step. I think, yeah, some way of getting the blood sample certainly to the lab or the Olink service provider, certainly in this iterative process, I think will be vital in the way forward. And then it could even be, you could think a screening program if you could somehow get a blood sample to a lab that you would step out. So yeah, very interested in seeing that. And I know that Olink, so I think, did the person, is it you from Olink asking that? I know that Olink have used their sample, have used dried blood spots or got results from dried blood spots. So yeah, I would be very interested. And I think that's a really good thought.
Excellent. Let's see here. What is the plan for the identified protein biomarkers? Will they progress to the next stage of development, such as laboratory developed tests or even in vitro diagnostics?
Yeah, so what we're really interested in is, again, doing iterative validation, certainly in high disease incidence areas, and we're in the process of setting up a collaboration with a research network consortia in India. And so we hope that with Olink and help, we can do some of these iterative steps and progress this into, well, potentially a laboratory developed test. But yeah, we're definitely advancing it in terms of exploring the diagnostic performance in an area where they could be of most clinical use. So India is the, so to speak, TB capital of the world. That it suffers, I think, a quarter of the world's cases, and it suffers the biggest proportion of the world's mortality to the disease. So the patients there could really do with some kind of screen that could identify this and actually find these millions of patients and try and reduce transmission. Because TB is ultimately treatable. I mean, the majority of the disease is drug sensitive. We have drugs that can treat it. Yes, there are some drug resistant cases, and these, but ultimately it's a treatable disease. And if you can diagnose it earlier, then you can have massive benefits all around.
Yeah. Are there any plans to perform the assay on a wide range of other pulmonary and non-pulmonary disease patients?
Yeah, again, I would love to. So I think testing it in so we know that combinations of markers perform differently with different in different geographical populations. So that's been shown with other studies, and why a diagnostic panel has to be tested in the panel of usage. I think the diagnostic performance also depends on what other diseases are prevalent in that population. And actually, if you're getting lots of other lung destructive diseases in that population, it would be important to test it against them from a diagnostic performance perspective. I think also, I think some of these proteins are related to lung destruction from the gene ontology work I've done, and not actually much is understood about them. And I think they may well be reflective of lung destruction. So evaluating their performance in something like lung fibrosis or COPD or other pulmonary conditions would be really important. And again, something I'd love to do. It's just a question of setting up the studies, and it's not, because as I alluded to before, pre-analytical variation is really important. So actually all these samples have to be collected in the same kind of study. It's no good just taking some samples from COPD patients, some samples from one study, some from another study, some from another study, and then looking for differential expression, because you never know whether that's due to the disease or whether it's due to pre-analytical variation or other factors that you haven't controlled for. So to set up a study to explore that, then you are talking big numbers, but it's something I would love to do.
Got time for a couple more questions. So we'll go with, do you have an idea of the performance of your 6-protein test compared to both IgRA for latent TB and a combination of the sputum PCR and direct examination for active TB?
So I think we've covered the latent thing before. I haven't got the direct comparison for that compared to a combination of sputum and direct examination. Yeah, so it's really interesting. So I think using it in combinations with other things is interesting. I mean, ultimately, you can ask people about their symptoms. You can examine them for TB, and then you can do the tests and still not get an answer. So we haven't got that direct comparison, no. But I am very interested in, as I alluded to, how host factors might alter the biomarker performance, and if they could be combined to give, if we're thinking these biomarkers could be used in a triage capacity, can you combine them with host markers to increase sensitivity, for example? Might that up the likelihood of your test and the patient subsequently having TB, because you've taken into account other things such as the patient's BMI, gender, smoking status, for example? Will that help? But no, we don't have the direct comparison.
Are the other respiratory infections mostly acute or chronic?
Yeah, good question. They were a mix, mostly acute. They were patients that had been admitted to hospital, so they were all patients that needed hospital admission from the mimic cohort. The ones that we had the microbiological diagnosis for, they were a mix of strep pneumoniae, staph pneumonias, influenza, so a mix of bacterial and viral, mostly more towards the acute than the really chronic end of the spectrum. But the important thing is there were people that presented with similar symptoms.
All right, we'll do time for one more. Anything else that gets sent in during the on-demand period, or anything we didn't get to today, we will be able to follow up with you afterwards. So we'll go with this next one here that was here. What is the sensitivity specificity of the assay in both bacterial yes, yeah. And negative samples? Apologize.
Yeah. So they were all bacteriologically positive. So the reference standard in all the studies was mycobacterial positive. So we haven't got that comparison. I mean, yeah, there's a different discussion to be had about reference standards when you're evaluating a diagnostic, but it was all compared to microbiologically confirmed infection.
That looks about all the time we have for questions today. Do you have any final comments you'd like for the audience?
Thank you very much for listening and for your interesting questions and discussion. As Matt said, I'm very happy to answer more, offline, or be approached offline. And obviously, if anyone else has any comments or has experience in similar research, and certainly has any experience in diagnostic technology or things that you think could be applicable to help the patients, then I'm really grateful to hear from you. I think all my contact details will be put out by the webinar details.
All right. Thank you again, Dr. Schiff, for your time and your important research. We'd like to thank LabRoots and our sponsor, Olink, for underwriting today's educational webcast. Before we go, we'd like to thank the audience again for joining us and their questions. Any questions we did not have time for today or that are submitted during the on-demand period, They'll be addressed by the speaker via the contact information you provided when you registered. This webcast can be viewed on demand. LabRoots will alert you via email when it's available on demand. We do encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you very much and goodbye.